All right. So some other terminology: scarification, to surface scar the seed. You're literally scarifying or scarring the seed, either physically or chemically. And you're overcoming the physical dormancy of that seed because it can't get water in. It can't start growing, even if you provide water, warmth, and oxygen and sunlight in the quantities that the plant needs. That seed is still just sitting there because the water can't get inside. And so, how do you do it? There are three methods: mechanical abrasion. So, I've done two different ways. One is using a coarse grit sand or fine grit gravel, depending on how you look at it. I don't remember if it technically was sand or gravel. 2 millimeters is technically the cutoff point for sand. But I would put the seeds in a bag with this coarse grit material, throw them in the dryer on zero heat, and tumble them for a set length of time, a relatively short amount of time. Take them out, and that tumbling with the grit was enough to get the seeds to be scarred enough without turning them to paste, without killing the seeds. So it took some practice to figure out how many minutes was the right time in our setup. And I'm not even going to bother giving you a time because you have to figure it out on your own. You have to play around to find out with your setup what works. But it would increase our germination from 20% to 85% or something with seeds that needed to be scarified. Another was just a piece of sandpaper. When I was using maple seeds, maples have a very hard coat. So I'd make sure I knew what I was doing. I was looking at the right side of the seed, and I would take the seed and drag it across the paper away from the embryo. So I didn't hurt the embryo, but I would just drag it across the sandpaper just a little bit, just this much, and stick it in the soil. That was all it took. That was enough to kind of break off a piece of the seed coat. Hot water is another type, and so you can put the seeds in hot water for a very short period of time. You don't want to kill the seeds. You just want to cause some sort of change in that seed coat where now water can get in. So you put them in the hot water for a period, pour that hot water through a strainer, run the strainer under cool water to take that heat away so it doesn't continue cooking it into the seed. Once you cool it, you plant it and start growing it. Acid treatment, I used to use that for tomatoes a lot because I was dealing with Galapagos Island tomatoes off the west coast of northern South America. And the Galapagos Islands have a very wonderful relationship between the tortoises that live there and the wild tomatoes. And the wild tomatoes are super sweet, small, smaller than typical cherry tomatoes, bigger than the typical grape tomatoes for some of them. It depends. There are a number of varieties that live there. And the tortoises would eat them. But if it was typical tomato seeds, they would not only digest the tomato, they would digest the seeds and get the nutrients out of the seeds as well. But the Galapagos Island tomatoes have very tiny seeds that are like hard grains of sand. And by going through the gut of the tortoise would then go to the bathroom, poop out the tomato, digested tomato, along with the chemical scarified tomato seeds. They weren't digested. They were just softened just enough where they would start growing. So that now tortoise poop would be moist, fertilized, and softened seeds, and the tomatoes would then start to grow right there. Lots of examples of that happening with birds and reptiles and mammals that eat different types of fruits and the seeds with them. And when they go to the bathroom, they poop out these scarified seeds ready to go. And so they've developed this kind of symbiosis between the plant and the animal, where the animal helps plant and spread the seeds. Now, we didn't have tortoises, and it would take too long to feed the tortoises and follow them around scooping their poop. So instead, we would take the tomatoes and put them in either uh, an acid treatment or possibly a bleach treatment. Now, that's just the opposite, but bleach did the same thing. Using a bleach concentration, again, we would soak them for the specified amount of time, run the material, the acid or bleach solution with the seeds, pour it through a strainer, and then run it under clean water for several minutes to get all of the excess acid or bleach off the seeds, and then plant them. And with some of those research seeds I was working on at UC Davis, the seeds from the Galapagos Island, uh, I would get anywhere from 10% germination before scarification up to perhaps 85, 90% germination after scarification. Huge difference in success. Now, that's still not nearly good enough for commercial seed. You need 99 or 100% germination for commercial seed. But with a little bit of work breeding the wild sweet tomatoes from the Galapagos Islands with the um, more traditional commercial tomatoes, we came up with a number. I had maybe, I don't know, 30 successful lines out of the 120 that were finalists, I would say. Um, Maybe 30 of them that were really good tomatoes with commercially viable seeds, different sized fruits, some of them extremely sweet, almost like candy. Wonderful, wonderful tomatoes. So that's the scarification. The next one is stratification, and that is using a temperature control or a temperature process to break dormancy. It's a physiological dormancy. So before I get into explaining stratification, imagine you are a dogwood tree living in Yosemite. Nice place, lucky tree. You're living in a beautiful Yosemite Valley. And it gets cold in Yosemite, right? So what happens? You are a tree, your flowers open in the spring, and then you get pollinated by um, the local insects, and then you develop these wonderful seeds that fall on the ground in late summer, early fall. And then maybe you have this nice warm rain come in a Indian summer, as they sometimes call it, in the fall, and it's beautiful, warm, and moist, and the seed starts growing. What would happen? The next thing this little tiny dogwood seedling would see is winter. The seed's this big. It just started growing. And that seedling is not going to live through winter. So instead, very few seeds would have been triggered by this nice warm fall rain. They need to be stratified first. They need to experience winter. They have a physiological dormancy with a cold sensitive chemical inside. And that cold sensitive chemical prevents the seed from growing, it keeps it dormant. But as soon as that seed has experienced a couple of days of winter, real cold winter, it now can start growing as soon as it gets warm again. So it experiences winter, that chemical breaks down over a few days. And now that it's winter, it's not going to be summer again or spring again for a while. You've got to get all the way through winter. So the seed's just sitting there. It's no longer physiologically dormant, but it's quiescent now. It's just waiting for the right time. Then it warms up in the spring and that seed starts to grow. So the seed needs the trigger of winter to break down those chemicals that are keeping it dormant. And once those chemicals are broken down, it can start growing. That is called stratification. Going back to the maple seeds, I needed to scarify and stratify my maple seeds for most of the maple breeding. And so what would I do? Scarify first with a sandpaper, making sure to only scratch the side away from the embryo. Stick it in a little bit of sterile soil. Then put the research seedlings or seeds that I just planted into the refrigerator and cool them down to the appropriate temperature. Leave them there for um, a week or two, depending on different trials, different varieties that I was working with. Then take them out, and they would start growing. So the scarification allowed water in. The stratification broke down the chemicals that were inside, saying, "Don't grow yet." And there are examples of plants. I've never dealt with them, but warm stratification might be necessary. Viburnum or red bud is something that actually uh, grows here in California. You could look up red bud viburnum. Beautiful plant. Um, but anyway, I won't talk about viburnum. But I'm assuming that when the seeds of viburnum come off, that they need to experience the heat so the seed doesn't start growing in um, a random warm period followed by drought or summer. It needs heat followed by cooler temperatures where it can germinate and be happy. So again, it's the same idea, just a little different. So we're talking about germination. There are actually two different types of germination. One is called epigeous germination and one hypogeous. Epi means on top of, hypo means underneath. Epi on top, hypo like a hypodermic needle goes under the skin when you get a shot at the doctors, right? Or a vaccination. Hypo. So epigeous germination means that the cotyledons or the seed leaves, they're not really leaves, but they are the leaf-like structures that have extra nutrients and food inside the seed. So when I'm eating a peanut, for example, the main part of a peanut that you eat is the cotyledons. That's the storage, the energy, the calories, the food. The embryo is just this little teeny tiny thing inside of that. And with a pea or excuse me, a peanut, um, those are going to stay as like gas tanks. If I grow a bean, which is closely related, but you know, kidney beans, lima beans, chickpeas, whatever, garbanzos, when they start growing, the beans push the cotyledons above the ground and they open up and they look kind of like leaves. Some species of plants, they look really a lot like leaves, others not so much. But epigeous means the cotyledons come up above ground. Cherries, beans, including soybeans and radishes, lettuce, spinach, the cotyledons come above ground. Hypogeous germination, the cotyledons stay with the seed underground, and only the true leaves, the actual plumule of the embryo, come up, leaving the cotyledons underground behind. And they don't look like leaves. They are just these little gas tanks of food underneath the ground. And so peaches, peas, and apricots are examples of hypogeous germination. So here's some drawings of those. Epigeous, here's a cherry. Pea would look exactly the same. The seed starts to grow. The hypo, or below the cotyledon, hypocotyl, is the part below the cotyledon. That is the stem below the cotyledon, as well as the embryonic root structure. Then the embryonic root structure develops a root system. And this is now the hypocotyl, the portion below the cotyledons, epicotyl, the portion above the cotyledons. And here you can see the cotyledons. They're kind of leaf-like. They're probably not going to be orange. That's just the artist's rendition of it. And then these are the true leaves. In hypogeous germination, like this peach, same starting, you've got a seed, the seed starts to grow, the radical emerges, the radical is the embryonic root, the plumule starts to push up, the plumule is the embryonic true leaves, and you still have hypocotyl, a portion of stem just below the cotyledon, and the epicotyl, the portion of the stem above the cotyledon, and the true leaves, and the cotyledon stays underground. Hypogeous and epigeous germination. And I am going to take one more break here.